the bacterial culprit behind sea star wasting disease. Vibrio pectenicida FHCF-3 explained. Imagine a silent predator lurking in the ocean, responsible for one of the most devastating diseases to strike marine life. Vibrio pectenicida strain FHCF-3 is at the center of this ecological nightmare as a key causative agent behind sea star wasting disease, a condition that has left researchers scrambling to understand its origins and impact. This disease doesn't just affect individual sea stars. It threatens entire ecosystems by decimating populations of these vital predators. But here's where it gets controversial. While some experts point to bacteria like Vibrio pectenicida as the main culprits, others argue that environmental stressors or viral infections might play an equally significant role. So, what's really driving this crisis? Let's dive deeper into the science and data surrounding this pressing issue. For those eager to explore the underlying research, detailed metatranscriptomic and 16S rRNA gene sequence datasets are publicly available through the NCBI Short Read Archive under bioproject number PRJNA 1195080. https www.ncbi.nlm.nih.gov/bioproject. Additionally, if you're interested in studying the genetic blueprint of Vibrio pectenicida strain FHCF-3, its whole genome sequence can be accessed via the NCBI GenBank repository with accession number JBLZMR0000. https www.ncbi.nlm.nih.gov/newcore. Raw sequence reads related to this genome are also archived within the NCBI Short Read Archive under bioproject number PRJNA 1232168. https www.ncbi.nih.gov/1232168. Furthermore, the complete 16S rRNA gene sequences of this particular bacterial strain have been deposited in the same repository under multiple accessions ranging from PQ 700,178 https://newcore PQ 7178 to PQ 763229 https://newcore PQ 763229, providing robust resources for further investigation. What about the tools used in this groundbreaking analysis? If you're curious about replicating or building upon the computational methods employed in this study, the code generated during the research process is freely accessible via Dryad at https://doi.org/10.5mkkWh7g9. This resource includes all scripts necessary to reproduce the findings presented in Reference 41, https://www.nature.com/articles/S41559-025-797-2. Now let's zoom out and consider why this matters beyond just academic interest. Sea star wasting disease has wreaked havoc on coastal ecosystems across North America, wiping out massive numbers of ochre stars and sunflower sea stars alike. For instance, studies such as Eisenlord et al. Work highlight how factors like population size structure and water temperature exacerbate mortality rates during outbreaks. Eisenlord M.E. Philosophical Trans. R. Soc. B. 2016. Meanwhile, Hewson et al.'s landmark study identified a denzavirus linked to mass mortality events, adding another layer of complexity to our understanding of the disease. Hewson, I. Proc. National Academic Sci. USA 2014. And yet, despite decades of research, many questions remain unanswered. Are we missing something critical in how environmental changes interact with pathogens? Could conservation efforts inadvertently favor certain microbial communities over others? This brings us to perhaps the most overlooked aspect of the story: the broader implications for biodiversity and ecosystem health. Dawson et al.'s recent review underscores the profound consequences of prolonged sea star declines, noting shifts in species composition and potential cascading effects throughout marine food webs. Dawson M.N. Bile Bull, 2023. Moreover, Harvell et al.'s alarming report connects rising ocean temperatures with increased susceptibility to disease among pivotal predators like Pycnopodia helianthodes, suggesting climate change could amplify future crises. Harvell C.D., Sci Advents 2019. These insights beg the question, are we doing enough to protect vulnerable species from emerging threats? To complicate matters further, there's growing evidence that microorganisms residing at the animal-water interface may actively contribute to disease progression, according to Aquino et al.'s findings, Aquino CA, Front Microbial 2021. Such revelations challenge traditional notions of pathogenesis and raise intriguing possibilities for intervention strategies. Yet, they also spark debate over whether focusing solely on specific pathogens like Vibrio pectenicida overlooks other contributing factors. As we reflect on these developments, it's worth asking, do you think current scientific efforts are sufficient to address the multifaceted nature of sea star wasting disease? Or should more emphasis be placed on interdisciplinary approaches that integrate microbiology, ecology, and climate science? Share your thoughts in the comments below. We'd love to hear your perspective. Starfish apocalypse. Scientists solve mystery of billion-dollar die-off. Imagine a world where a crucial part of our ocean's ecosystem is on the brink of collapse. This is the stark reality we're facing, as billions of starfish, also known as sea stars, have perished in a decade-long epidemic that has left scientists baffled. But here's the astonishing part. After years of research, scientists have finally cracked the code and discovered the cause of this mass die-off. The culprit? A type of bacteria that has also infected shellfish, according to a groundbreaking study published in the journal Nature Ecology and Evolution. This epidemic has ravaged over 20 species of sea stars, with the sunflower sea star being the worst hit, losing a staggering 90% of its population in just 5 years. The disease, known as sea star wasting syndrome, is characterized by gruesome symptoms, including lesions, twisted arms, and eventual disintegration of the sea star's body. Marine disease ecologist Alyssa Gaiman describes the scene as really quite gruesome, with healthy sea stars having puffy arms sticking straight out, but those infected with the disease suffering from lesions and arm loss. The study's findings have solved a long-standing question about a very serious disease in the ocean, according to Rebecca Vega-Thurber, a marine microbiologist. So, what took scientists so long to identify the cause? The answer lies in the complex and challenging process of tracing the source of environmental diseases, especially underwater. Early research hinted at a virus, but it turned out to be a false lead, and other efforts missed the real killer due to the limitations of studying tissue samples. However, the latest study includes a detailed analysis of the coalomic fluid, where the bacteria Vibrio pectenicida were found. This breakthrough has significant implications for the conservation of sea stars and the entire Pacific ecosystem. The International Union for Conservation of Nature has listed the sunflower sea star as critically endangered, and scientists are now exploring ways to intervene and help the remaining sea stars. This could involve testing which sea stars are still healthy, relocating them, or breeding them in captivity to later transplant them to areas that have lost almost all their sunflower sea stars. But here's where it gets controversial. The study notes that Vibrio bacteria have been linked to climate change, as they thrive in warming water temperatures. This raises important questions about the relationship between rising seawater temperatures and sea star wasting disease. As scientists delve deeper into this issue, they may also explore whether some populations have natural immunity and if treatments like probiotics can help boost immunity to the disease. The stakes are high, not just for sea stars, but for the entire ecosystem. Sunflower sea stars play a vital role in maintaining the balance of the ocean, as they devour excess sea urchins that can destroy kelp forests. With many fewer sea stars, the sea urchin population has exploded, leading to the destruction of around 95% of the kelp forests in Northern California within a decade. These kelp forests are crucial, providing food and habitat for a wide variety of animals, including fish, sea otters, and seals. Researchers hope that the new findings will allow them to restore sea star populations and regrow the kelp forests, which are often referred to as the rainforests of the ocean. And this is the part most people miss. The recovery of sea star populations is not just important for the ocean's ecosystem, but also for the many species that depend on it. So, what do you think? Can we reverse the damage and save the sea stars? Should we prioritize the conservation of sea stars and the restoration of kelp forests? Share your thoughts and join the conversation. Scientists discover bacterial cause of sea star wasting disease. Imagine a world where billions of sea stars, once thriving in the ocean, suddenly begin to wither away, their bodies disintegrating into a gooey mess. This is not a scene from a science fiction movie, but a harsh reality that has been unfolding along the west coast of North America for over a decade. The culprit behind this devastating disease, known as sea star wasting disease, has finally been identified, and the discovery is sending shockwaves throughout the scientific community. Researchers at the University of Washington and the Hawkeye Institute in British Columbia have cracked the code, revealing that a strain of bacteria called Vibrio pectenicida is responsible for the massive die-off of sea stars. This breakthrough finding is a major milestone in the quest to understand and combat this disease, which has ravaged sea star populations from Alaska to California. The sunflower star, a species that was once abundant in the region, has been particularly hard hit, with over 5 billion individuals succumbing to the disease. This has had a ripple effect throughout the ecosystem, as sea stars play a crucial role in maintaining the balance of the ocean's delicate food chain. The loss of these predators has allowed sea urchins to proliferate, leading to the destruction of kelp forests and the loss of biodiversity. But here's where it gets even more complicated. The discovery of Vibrio pectenicida as the primary cause of sea star wasting disease is not without controversy. Previous studies had pointed to a virus as the culprit, but these findings were later disputed. The current study, published in the journal Nature Ecology Evolution, provides conclusive evidence that Vibrio pectenicida is the primary cause of the disease. So, what makes this study so convincing? For starters, the researchers took a more comprehensive approach, analyzing not just the external tissues of the sea stars, but also their internal fluids. This allowed them to isolate the Vibrio bacteria and demonstrate its role in causing the disease. But the journey to this discovery was not without its challenges. The researchers had to develop a sophisticated system to avoid microbial contamination, using copious amounts of ethanol, bleach, and betadine to keep their equipment and samples clean. They also had to work with a limited number of sea stars, as the species is now critically endangered. The emotional toll of this research should not be underestimated. Studying a disease that is ravaging a species on the brink of extinction can be emotionally draining, especially for the research assistants who works tirelessly to care for the sea stars. As one researcher noted, "It's a traumatic experience whenever our stars get sick." Despite these challenges, the researchers remain optimistic about the future. Now that the cause of the disease has been identified, they can begin to develop strategies to combat it. This includes the development of probiotics to help healthy sea stars ward off the disease, as well as testing other species to see if they are also susceptible to Vibrio pectenicida. But as we celebrate this breakthrough, we must also acknowledge the uncertain future of labs like the Marrowstone Marine Field Station, where much of this research was conducted. The proposed budget cuts to the U.S. Geological Survey threaten the very existence of these labs, which are crucial to our understanding of the ocean and its inhabitants. As we move forward, it's essential that we prioritize funding for scientific research and conservation efforts. The fate of the sunflower star and countless other species hangs in the balance, and it's up to us to take action. So what do you think? Should we prioritize funding for scientific research and conservation efforts? Share your thoughts in the comments below.